Hello everyone, welcome to Food Science. This lecture is about the food science of carbohydrates, and this is the first video. These are the learning objectives. At the end of this lecture, students should be able to describe the physical properties of carbohydrates and their role in food systems, describe the chemistry of carbohydrates and their role in food systems, list the applications of carbohydrates in food industry, describe the methods of carbohydrate analysis, describe the types of sweetness. Before we proceed into this lecture, there are prerequisites. Students should have knowledge on the following aspects: carbohydrate classification into monosaccharides, disaccharides, oligosaccharides, polysaccharides, and other sugar derivatives. The structures of these carbohydrates, something you should know about the glycosidic bonding, and the food sources of these different carbohydrates. These are two textbooks recommended for you to read. Both these books are available in the Griffith Library, and I have given you the chapters and the relevant pages that you should read. Talking about carbohydrate biosynthesis, photosynthesis is a natural process which is occurring in plants and produces glucose, which, as we know, is a carbohydrate. And this slide shows some introduction to carbohydrate classification, which is also already known by all of you. Carbohydrates can be classified as monosaccharides, which contain only one sugar unit, disaccharides, which contain 2 sugar units, oligosaccharides, which contain 3 to 10 sugar units, and polysaccharides, which contain many sugar units. Let's look at the physical properties of carbohydrates. Physical properties depend on several factors like the monosaccharide composition of the carbohydrate, glycosidic linkages. So this diagram on the top shows a glycosidic bond, and then depends on the functional groups like the aldehyde groups, ketone groups, and hydroxyl groups, the molecular size, and the presence of branching. So the first physical property that we talk about is the solubility. Unmodified monosaccharides, disaccharides, and oligosaccharides are generally soluble in water. In this diagram below, you can see these carbohydrates, they can form hydrogen bonds with water so that they are soluble in water. However, highly crystalline polysaccharides like cellulose, or gelling polymers like agarose, or highly branched cross-linked polymers like starch, these are insoluble in water. The next physical property we look at is the hygroscopicity. Hygroscopic nature is the ability of these sugar molecules to uptake water and retain the moisture. So this also varies with several factors like the structure of the sugars, the isomers they have, and the sugar purity. Isomers is a term that you already know. Isomers have identical molecular formula, but they have different arrangement of atoms. For example, glucose, fructose, galactose, they have the same molecular formula, C6H12O6, but the arrangement of atoms is different. So those are the isomers. So when sugar powders or sugar granulates, when they cake together, their solubility decreases. Caking together means usually these sugars have free-flowing granules, but caking means that they come together forming a lump, and this is dense. In this dense structure, in this lump, the solubility is lower. If you want to know why this happens, because now it's densely packed and it reduces the surface area that is available to interact with the surrounding water. So now the solubility is decreased. And then these sugars have the ability to retain the moisture like this. They can attract water and retain that. This is used in bakery industry to make the food more soft, because the sugars can retain moisture. that makes the bakery products more soft, giving them a moist, soft texture. The next physical property is the optical rotation or the optical activity. The optical activity is the ability to rotate a plane polarized light. So if you can see the diagram below, the ordinary light that rotates or that vibrates in several directions, but if we send this through a polarizing prism, we can obtain plane polarized light, which is vibrating only in one direction. And if we pass this light through a sample which contains sugar, this changes the direction of rotation. With that, we can identify the type of the carbohydrate, the purity of the carbohydrate, and also the concentration. So this is used in food industry and pharmaceutical industry. Next, I'm going to introduce you to the starch granule. You already know about this, but just refreshing this knowledge is important when we go to the next few slides. So starch is a polysaccharide which is present or stored in the roots, seeds, and endosperm of the grains, and this is insoluble in water. So the starch, the monomer of the starch is glucose, and the glucose is bound to each other, and this can be a linear structure like in amylose. They have only alpha-1 for glycosidic bonds and it forms a linear structure, or this can be a branch structure like in amylopectin, which has both alpha-1 for glycosidic bonds and alpha-1,6 glycosidic bonds. So this starch granules contain both amylose and amylopectin in different ratios or different percentages. For example, the starch coming from potatoes, tapioca, they have more amylose, and the starch coming from rice, wheat, they have high amylopectin content compared to amylose. So if the starch has a high amylose content, like high linear polymers, they are able to form a gel, like they can incorporate water and they can form this three-dimensional structure, a semi-solid structure. This is called the ability to gel. But if it does not have enough amylose, they cannot form this gel. Instead, they will be more thick, more viscous. So whenever there are high amylopectin, it becomes more thick, more viscous paste, which means that they cannot form a gel. Now let's talk about the food chemistry of carbohydrates. The first reaction is a reduction to sugar alcohols. As you can see in this diagram, it shows glucose, and this carbonyl group can undergo reduction and it forms different sugar alcohols, which are also known as polyols. So the examples of some sugar alcohols are xylitol, sorbitol, mannitol, glucitol, etc. And then these sugar alcohols are used in food industry to decrease the water activity, which means these sugar alcohols can attract water and that decreases the amount of water that is available for reactions. And also that act as softeners. That is again because they can attract water and they can inhibit crystallization, improved rehydration characteristics of dehydrated food due to the same reason that they can attract water. And then these sugar alcohols provide fewer calories compared to the sugars. And another important aspect is that these sugar alcohols are not readily fermented like sugars. Therefore, food industry uses this to produce chewing gums. Like in chewing gums, this has to retain in the mouth for a longer duration. So these sugar alcohols, they do not undergo fermentation readily, so they can be kept in the mouth for a longer duration. The next reaction is the oxidation. So you can see here in the diagram, glucose undergoing mild oxidation, and this carbonyl group undergoes the oxidation and it produces gluconic acid. While producing gluconic acid, they produce gluconodelta-lactone as an intermediate. This gluconodelta-lactone can be used as a leavening agent in baked food, raw fermented sausages, or dairy products. And the other thing is glucuronic acid, where the carbon number 6 undergoes oxidation and it produces glucuronic acid, or these uronic acids are important as gel forming and thickening agents. For example, pectin, which is a uronic acid, D-galacturonic acid, and seaweed-derived alginic acid. These are used as thickening agents in food industry. The next reaction we talk about is the Maillard reaction or the Maillard browning. Whenever I say Maillard browning, what comes to your mind should be this bakery products. The brown color, the browning that is present in bakery products, that is a result of the Maillard reaction. So for this reaction to occur, it needs carbonyl carbon of a reducing sugar, for example, glucose, fructose, maltose, lactose. And then there should be an amino group coming from a protein or peptide, amino acid, or an amine. So these two things react, and this readily occurs at high temperature, low water activity, and with longer storage period. So here you can see the reaction that is happening here. Sugar molecules react with the sugar, CHO group reacts with the NH2 coming from the protein or any other peptide or amine compound, and that undergoes several reactions, and in the end it produces melanoidins. This gives the brown color. This is the brown color that you can see in the bakery products. And also this produces different volatile compounds giving the aroma. The fresh baked products are having a specific aroma, or the aroma that is produced during roasting, frying, that is produced during as a result of Maillard reaction. And also that produces flavors, like the flavors, the distinctive flavor that of chocolate, and also the bitterness you get in coffee, or sometimes the off flavors that we observe when we grill fish or meat. And also it produces another compound called reductones, which contributes to the stabilization of food against oxidation. And the other disadvantage of Maillard reaction is the loss of the nutritional value. It causes the loss of essential amino acids. So whenever there are sugars and amine groups, CHO group and NH2 group, they react under the conditions like high heat, undergoing this Maillard reaction, and this amine group is now used for the reaction, therefore it causes the loss of essential amino acids. The next reaction is caramelization, which is also producing a brown color, but this is different to Maillard reaction because, as I told you, for the Maillard reaction to happen, we need both carbohydrates and amine groups. But caramelization happens in the absence of amine compounds, just the sugars are undergoing this reaction. So you might have seen this when you are heating sugar in a pan. It looks like the sugar is just melting and then it's becoming brown, but it's not just melting. A series of reactions are happening within this food system. So sugar rings, they open and they lose water, and then they break down the existing glycosidic bonds, and then they form new glycosidic bonds. They combine with each other and then form larger molecules. As a result, they produce different brown colored products, and you get the typical caramel aroma. So this final product, which is brown in color and giving you the caramel aroma, can contain several chemical compounds like hydroxyl groups, carbonyl, carboxylic, enolic, and phenolic groups. So this caramelization happens in food items like beer, cola beverages, and you can see this caramel on top of caramel pudding and some confectionery items, and also in caramelized onions. The next reaction is the fermentation. Fermentation is a metabolic process which converts a carbohydrate or like a starch or sugar into an alcohol or an acid, and this needs an organism. It may be yeast or bacteria. So fermentation happens in bread, where this glucose, with the help of yeast, produces ethanol and carbon dioxide, and this carbon dioxide helps the bread dough to rise. And also this happens in vinegar, uh, using the acetic acid bacteria. Vinegar is acetic acid, so it um, undergoes the fermentation reaction and also oxidation. Then this also happens in fermented milk products where lactose is fermented to become lactic acid with the help of lactic acid bacteria. And due to the presence of lactic acid, the milk can coagulate and produce fermented milk products like yogurt. This is the end of this video. However, the lecture still continues. So in the next video, I'm going to talk more about the food chemistry of carbohydrates. Thank you.